Hi everyone, welcome to this presentation called A Genetic Seminary. This will be narrated by Angelica Ralgosa and me, Camilo Shaw. Well, but what is biodiversity? The main concept about biodiversity was elaborated in 1992 by the Convention on Biological Diversity, CBD. The variability of living organisms from any source, including, among other things, terrestrial ecosystems and marine and other aquatic systems, and the ecological complexes of which they are part. It includes the diversity within each species, between species, and ecosystems. The girl in the photo of the left is about Severn Suzuki, who talked about the relevance of caring for biodiversity for future generations. Hierarchy? Yes. The biodiversity is made up of three closely related elements. This table, made by Global Biodiversity Assessment, shows a kind of hierarchy where ecological biodiversity or space is the most relevant element because it contains the physical environment that is where biological interactions develop. Examples: biomes, bioregions, landscapes, ecosystems, habitats, and populations. Second, genetic diversity, which includes the components of the genetic code of each organism. Examples: populations, individuals, chromosomes, genes, nucleotides. Finally, the diversity of species or organismal diversity, which are living beings with common characteristics. Example: kingdoms, phylum, families, genera, species, subspecies, and populations. Although measuring biodiversity is a complex task, species richness, that is, the total number of species present in a given place, is considered one of the most appropriate indicators to measure biodiversity. One example is this investigation: Biodiversity of Colombia, Significance and Regional Distribution, carried in 2015, is an inventory of biodiversity according to the geographical regions of Colombia. Thus, it can be seen in the graph that the Andes region concentrates the greatest vegetation richness, while the Paramo has the greatest variability. Most likely, it is due to the influence of the physical environment and environmental conditions. Genetic biodiversity. So, our interest is focused on biodiversity at the genetic level. The photograph on the left shows the diversity of crop species, and on the right, the potato diversity. What? How? Where? The diversity found within species is the fundamental basis of biodiversity at higher levels. Genetic variation determines the way a species interacts with its environment and with other species. All genetic diversity arises in the molecular file and is closely linked to the physicochemical characteristics of nucleic acids mutations. But How it is possible to quantify diversity at the genetic level? First, genetic variation can be detected on a molecular scale by directly studying the change in DNA structure or indirectly in the proteins that code specific genes. To quantify the genetic variation within populations, molecular data is used to determine the first, average level of heterozygosity, second, the proportion of lossy polymorphic, and finally, the total or the average of the number of alleles per locus. There are many ways to evaluate the genetic variations. For example, measure the polymorphisms two or more types of DNA among individuals of a species. These polymorphisms can be detected by a wide variety of techniques that reveal sequential changes in small specific regions of the DNA. Randomly expanded polymorphisms, or RAP, which refers to unique segments of DNA that are identified by primers and then expanded through a polymerase chain reaction, or PCR, to reveal differences between individuals. So, you show this graph that illustrates the methodology of quantification of biodiversity at the genetic level via RAPD. Here, two samples of the organisms to be evaluated are taken. These sections of the genetic code are amplified through universal primers, and in this way the difference in the genetic traits between them can be appreciated and analyzed. Finally, to complement the last explanation about RAPD method, we have this study done in Uruguay in 2004 uses the previous technique of quantification of biodiversity at the genetic level to evaluate 39 varieties of Solanum commersoni and 2 varieties of common potato Solanum tuberosum. This dendrogram shows that big variety between the wild Solanum species or varieties and the common potato (Solanum tuberosum). This allowed them to conclude the existence of high genetic variability in the wild varieties evaluated, opening a big possibility for plant breeding applications.
How genetic variety affect biodiversity. To survive under various environmental conditions for decades, long-lived population require create a leading diversity. Da Costa, Carmelo, and Stevenoff 2015. El wall is one of the most important measures of the genetic diversity. It depends largely on the effective population size and past evolutionary history. Allele frequency. Allele frequency is the proportion that is observed of a specific allele with respect to the set of those that can occupy in a fixed position in a chromosome determined in the population is not experienced evolutionary change. Hard behavior law. It establishes that the genetic composition of a population remains in equilibrium as long as natural selection and no other factor and not mutation occur. Therefore, it does not generate evolutionary change. Natural selection. It is a phenomenon of evolution defined as the differential reproduction of the genotype of biological populations. From the moment there are differences in the reproductive success of the different genetic variants, there is natural selection. This disruptive selection, extreme ranged prevail, light and dark claims. Stabilizer selection, the Evers range prevails, the lizard with medium side tail. Directional selection, the trait prevail and on one end giraffe with the longest neck. Eliminate unique individuals from a population. Mutation. It occurs spontaneously and suddenly or by the action of a physical, chemical, or biologic agent, produce variants in the characteristic of the population. It is not necessarily transmitted to the offspring. It is the random change in the nucleotide sequence or in the organization of the DNA. Beneficial mutation usually goes unnoticed and feared so that the evolutionary advantages are slowly manifested. If the new mutated genetic provides biferi, it will gradually replace the original in the population. Although in the short term they may seem harmful, mutations are essential to our long-term existence. Without mutation, there would be no change, and with change, life will not evolve. How genetic organisms affect biodiversity? Transgenic food. They made it to incorporate genetic material genes from another organism into a plant. 1. In concept of taking a DNA fragment. 2. Obtained by restriction inside action. 3. Which validity run by means of the DNA leads enzyme to the vector of the plasmid generate a new molecule known as recombinant. 4. The vector use contains sequences that allow replate and sequences the facility in selection. 5. Sometimes there are genes that confer resistance to specific antibiotics. The combined DNA obtained in the introduced into microsome. 6. The one that is cultivated and selected for it resists to antibiotic. 7. Giving us an end the creation of the herbaceous and pesticide-resistant species. Cloning. From the birth of the Dolly sheep in 1996, the first one derived from an adult animal, Church Sanchez and Franco, 2004. Cloning technology that allows the application of the power of genetic engineering and genomics in animals, plants. And the biodiversity, new advances in cloning and their potential in genetic variation in livestock. In our view, the scientists issued for breeding programs live in whether close can increase genetic progress without across the biodiversity. Cloned adults may be an important route to protect biodiversity since it may allow for more genetic variation to be made and valued for the breed development in the future that is practiced at present. Elite variation within individuals. First, C is not generated at the advantage, but at losses where it is invented to the beneficial to use the cloning made exploit in more welded.